everyone, and welcome to today's webinar titled Advanced mRNA Vaccine Solutions by GenScript. This webinar is part of the 5th annual event in the Bioprocessing Virtual Event Series. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to genscript.com. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located on the left of your screen. You can also participate by submitting as many questions as you like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the right of the abstract. This survey will be available during the live and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Roumen Bogoev, Head of Segment Marketing, GenScript USA Incorporated. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Roumen, you may now begin your presentation.
Okay, thank you, Christy, for your introduction, and thank you, everybody, for attending our webinar on advanced mRNA vaccine solutions. Here is the agenda for our webinar. I'm going to go over quickly over types of vaccines, mechanisms of action. I'll discuss our mRNA vaccine solutions from GenScript. I'll briefly touch on other vaccines, protein, DNA, and virus, and what kind of solutions we have for those vaccines, and then with short conclusion. So first, before I go into vaccines, a little introduction about GenScript. GenScript is a global company with a lot of services and products. We were founded in 2002. We are publicly traded on the Hong Kong Stock Exchange. We have about 4,000 employees right now, and we are very well known to be #1 in gene synthesis. We also have multiple production sites. Some are in China, some are in US, some are in Singapore, and we have served over 200,000 customers worldwide. Our headquarters is in Piscataway, New Jersey, USA, and we offer 24-hour customer service. We have fast global logistics. We offer high quality and very innovative services and products. We have very diversified offerings. We have 4 different business units under GenScript. The Life Science CRO business unit, they offer a wide range of services. A lot of their processes are fully automated for consistency and efficiency, and they typically work in the REO to preclinical grade services. We also have the Biologic CDMO division, GenScript ProBio. They mostly work in the clinical and preclinical space. They have high GMP capacity to meet the market demand, and they have very strong biologics expertise. We also have the catalog products division. They offer high quality off-the-shelf reagents, very innovative instrumentation. Lately, they have been launching many instruments related to automation and automating various lab processes. We also have the synthetic biology business unit, BestZyme. They focus mostly on innovating food industry, and they have very broad portfolio. They have very strong R&D and application capabilities. My team, we focus on several different market segments. We focus on vaccines, gene therapy, cell therapy, antibody therapeutics, and IVD. And we work all across all these business units to assemble portfolios that are best suited for these market segments. So what is typical vaccine workflow and what are the typical vaccines that are on the market? According to the WHO website, there are 3 generations of vaccines. The first generation of vaccine was mostly virus-based. Those were either inactivated virus or live-attenuated virus. Then the second generation was mostly subunit vaccine, where antigen basically is extracted from the virus or is contracted by genetic engineering means. And those are typically peptide or recombinant protein vaccines or virus-like particles. And then the third generation vaccine is mostly nucleic acid based. So these are the newest vaccines. We saw the COVID-related vaccines, where most of them that came out first were nucleic acid based. So they would be DNA, mRNA, or nucleic acid packaged in a viral vector. So what is the typical vaccine mechanism? Typically, the antigen enters the cells, the human body, and then is picked up eventually by the antigen-presenting cells, and is usually converted, whatever the antigen shape was, DNA, RNA, or virus, is typically converted to protein first, and then digested to peptides. These peptides are incorporated to the MHC class 1 and class 2 complexes, and they're presented to the T cells. When T cells encounter these antigens, they get activated, and correspondingly, they create cellular immunity or humoral immunity. So this is the typical vaccine mechanism. And what are the different vaccine types by function? Typically, we have prophylactic vaccines that are usually used before the pathogen enters the human body. And there are also therapeutic vaccines that are typically used after the human body is infected by the pathogen. And some examples of prophylactic vaccines are the vaccines for Ebola, HPV, SARS-CoV-2. We all experienced that. And some of the therapeutic vaccines, they focus typically on the tumor-associated antigen or tumor-specific antigens. And they're usually cancer-focused. However, there are some vaccines that are also now starting to focus on infectious disease. So what is the difference between therapeutic vaccines versus immunotherapies? For example, antibody therapeutics, they belong to the passive immunotherapy group, which uses antibody drugs to target the antigens, and basically they would label the tumor cells or the infection cells, and they would basically attract the immune cells to kill them. Cell therapies are typically using engineered cells that make more efficient cells for killing. For example, T cells, NK cells are engineered to become better at finding the tumor cells and killing them effectively. And then therapeutic vaccines are typically used to educate the immune system to recognize and proactively kill tumor cells and more effectively find them and kill them. In a large number of therapeutic vaccines are currently in clinical and research stage. Therapeutic vaccines are expected to be very important complement to tumor immunotherapies. And a lot of the companies that actually developed the COVID vaccines, those were companies that previously were working on therapeutic vaccines, BioNTech, Moderna, CureVac, and hopefully they're going to be launching their therapeutic vaccines very soon. So what is typical vaccine development workflow? Typically, the development workflow starts with finding the pathogen, identifying it, analyzing the antigens that are related to this pathogen, and basically making decisions on what type of antigen will be used for the vaccine. For example, if you take the COVID vaccines, decisions have to be made. Should the spike protein be used for the vaccine, the RBD domain of the spike protein, combination of the spike protein with an N protein, and so on. The next step is usually deciding on what kind of compound is going to be used for the vaccine. Would it be protein, peptide, DNA, RNA, virus? optimization of this compound for efficiency, and then optimization of the delivery mechanism. Then typically preclinical studies are completed with animal models and looking at efficacy in animal models, immune response, side effects, and so on. And then the clinical stage starts with different number of patients, clinical trials, looking at the clinical efficiency and also the resilience and how long the immunity is proceeding. So going back to the workflow, what can GenScript do for antigen identification and selection? There are usually two different types of studies depending on the type of vaccine. Pathogen study for virus, usually the virus is isolated, identified, the virus structure is analyzed, the specific antigens are identified. Very often antigen epitope study is performed to understand the exact epitope for the antigen. For tumor studies, usually studies are focused on tumor-associated antigens. Those are usually highly expressed in a tumor and very low expressed in normal cells. Or tumor-specific antigens that are highly only expressed in the tumor. They're not expressed in normal cells at all. Tumor-specific antigens are usually the more efficient way because they come with less side effects. So what can GenScript provide for this antigen identification and selection? We have genome analysis products and services. For example, we can offer NGS enrichment libraries. Those are based on our GenTitan oligopool methodology. We can also provide PCR and qPCR related products. We have very strong portfolio of PCR enzymes, nucleotides. We can develop custom primers, custom probes, we can label them. On the protein analysis side, we can provide a lot of tools for protein detection and quantitation, basically Western blotting products, antibody catalog or custom developed antibodies, polyclonal, monoclonal, mouse, rabbit. We can develop custom ELISA kits. For analyzing the immunogenic response, we can usually offer products for epitope mapping, peptide libraries. We can also develop custom ELISA kits for detection. So going a little bit more into detail into the different services. So for example, for NGS libraries, we can develop hybridization-based libraries or amplicon-based libraries. They can be packaged in different formats. They can be in tubes or plates. They can be lyophilized, and we, of course, send them with the corresponding documentation. These libraries are based on our GenTitan on-chip synthesis technology. This technology enables us to synthesize up to 8 million oligos in parallel on single chip. And usually the coverage is 99.9%, so very high, and with 20 to 30 milligrams quantities of oligo. And we can also scale this down. We have smaller libraries that are 92,000 oligos, and they can also be developed very efficiently with 99.9% coverage and 5 to 10 micrograms of oligos. For PCR, we can offer a variety of products, as I mentioned, and we can synthesize the forward primers, reverse primers. We can synthesize the probes. We can label the probes for you. We can also deliver them in different formats, tubes or plates or lyophilized powder. We have also DNA enzymes, DNA polymerase enzymes. We have excellent anti-DNA polymerase antibody that allows companies to create hot start Taq polymerases. We have reverse transcriptases, DNTPs, and various plasmid vectors. For immunodetection portion, for protein analysis, we have complete portfolio of Western blotting products. We have excellent sure-page precast gels. Those are gels that run very fast. They're very high quality, very high resolution. We have the corresponding tanks for running them. We have pre-stain standards. We have the widest range protein standards on the market, from 5 kilodalton to 270 kilodaltons. We also have Western blotting products. We have eBlot protein transfer system, which is very efficient wet blotting transfer. is fast and easy to use, and at the same time gives you the ability to very efficiently transfer your proteins with wet blotting. This can be bundled together with our easy waste automated membrane detection system that does automatically the blocking, primary antibody incubation, secondary antibody incubation, all the washes required for that, and gives you a membrane that's ready for detection. For gels that are stained instead of used for Western blotting, we also have automated stainer destainer that can accomplish the whole staining destaining within about 10 minutes. We also have very high quality catalog antibodies that are off the shelf and ready to ship. In addition to the catalog antibodies, we can also develop any custom antibody. We can develop custom polyclonal antibodies, custom monoclonal, mouse monoclonal antibodies, custom rabbit monoclonal antibodies. We have multiple technologies for antibody development, including single B-cell isolation and using the Beacon platform for testing. And we can do also antibody labeling. If those antibodies are going to be used for flow cytometry, for example, or ELISA kits, we can also label them correspondingly for you. In addition to the antibodies, we can do antibody kit development. So we can develop various sandwiches ELISA, competitive ELISA, and so on. We can develop anti-drug ELISAs, anti-drug antibody ELISAs. We can develop custom HCP kit for your specific expression system and for your specific expression workflow. For immunogenicity testing, we can develop various peptide libraries. We have overlapping libraries, we have alanine scanning libraries, we have truncation libraries, and so on. Various types of libraries that can be very useful for testing. And they can come with different delivery dates, depending on the type of library that you're looking for and depending on the peptide sequence and length of the peptides. mRNA vaccines are very effective. As we can see from the COVID developments, they were developed very quickly and they seem to work very effectively. However, they have some challenges. Sometimes during application, they can cause cytokine storms. Some other side effects could be allergies, renal failures, heart failures, infractions. Therefore, they have to be optimized very carefully. They have to be very careful, the sequence, the structure, the delivery mechanism, the dosing, all these parameters need to be optimized in order for the vaccines to be effective and safe. mRNA vaccines are also pretty challenging to develop. mRNA is very well known for its stability, degrades very quickly. Therefore, a lot of optimization has to be done on that part. 3' UTR optimization, structure optimization, RNA length optimization. Shorter RNAs are usually more stable. Modified uridines have to be used sometimes to prevent degradation by enzymes. mRNA translation may need to be optimized, again, with the 5' and 3' UTR regions, 5' capping efficiency. The more complete, the more stable and more better translation efficiency. Poly-A tailing is also very important. That's well known, and codon optimization. Also, the RNA delivery mechanism needs to be determined and optimized. Will it be lipid nanoparticles, polymers, peptides, other means of delivery? So all these parameters need to be optimized in order for us to deliver a successful vaccine. So looking at the mRNA structure, we have the 5' region that usually needs to be capped to improve the stability of the mRNA, eventually lead to better translation. 5' and 3' UTR regions, they need to be optimized for stability and translation. Coding region needs to be optimized with codon optimization to improve the translation efficiency. The 3' region, usually poly-A tailing needs to be optimized to provide the stability and translation efficiency. So all these parameters need to be optimized. And what can GenScript do for your developments? Basically, at GenScript, we can go from the beginning to the end of this process. We have very good tools for codon optimization. We have already pre-optimized UTR designs that you can use. We can do the gene synthesis. We are very well known to be excellent at gene synthesis, anything from short oligos to very long DNA fragments. We can produce your plasmids. We can develop the plasmid, construct it, and do large preparations of the plasmid. We can do enzyme production if needed, and we can do the IVT mRNA synthesis with all the required modifications, optimization of the sequence, the UTR regions with the different uridine pseudonucleotides and so on. So going in a little bit more detail in some of those services, for example, for gene synthesis, of course, we offer a number of different gene synthesis services. We can do fast, standard, economy, gene brick. They vary depending on the size of the gene fragment that needs to be synthesized, the complexity of the gene fragment. So based on that, we would recommend different services. A gene brick is usually used for very, very large fragments that are very difficult to synthesize a single fragment. And we can synthesize them as multiple fragments. And we also have excellent cloning kit that would allow you later on to assemble these fragments back into your plasmid. We also offer web-based software for codon optimization that makes it very simple and easy. We also offer the GenSmart design tools for your DNA construct design and optimization. And also, of course, we can provide instant quotations on your service requests. So going a little bit more detail about the plasmid preparation services we offer. I'm not going to go over this data. I'm just going to point out that we have several different grades of service. We have service type for research grade, for basic research, industrial grade for early drug discovery, and preclinical grade for preclinical studies. And those vary based on the quantities we produce, the quality of the purification, and parameters we test in QC. And of course, all of this can be customized for your needs. Our GenScript ProBio CDMO division, of course, can take this into the clinical range. They can offer GMP production of your plasmids for mRNA vaccines, for DNA vaccines, and so on. They have different great services, plasmid CMC, preclinical, clinical, and then post-clinical, basically production of the plasmids. If you decide that you would like to keep some of these operations in-house, we also offer, as I mentioned initially, we have products division that produces a lot of innovative products and instrumentation. We can also offer you instrumentation that can help you do your own plasmid purifications. AMAC4 is basically out-of-the-box automation. It can use one controller and multiple purification modules. Each purification module can purify up to 6 plasmid samples. Up to 4 modules can be connected to one controller. So this can give you very good scalability. If initially you don't need this high throughput, you can get 1 or 2 modules and later on add more modules, and they can be seamlessly added to the system. The purifications are very high quality, very high yield, purity, and basically low endotoxin for expression for transfection level plasmids. We can also perform the IVT reactions and deliver mRNA product to you. This is here the usual mRNA structure. Usually the capping needs to be done on the 5' region. We have optimized this to achieve very efficient capping. And right now our efficiencies could be close to 99.5%, very high capping efficiency. You can see when we compare with some of the leading competitors on the market, we compare very favorably for that. So a little bit more detail on our mRNA service options. We can provide you scale-up options. We can produce micro, mini, and midi from 0.2 milligrams all the way to 20 milligrams of mRNA. We can also provide larger quantities, customized service. We can also, of course, do the capping options. I'm sorry. We can produce uncapped, cap 0, cap 1. We can produce different versions of poly-A tailing, no tail, 100A tail, customized tailing based on your requirements. We can introduce the modified uridines into the sequence structure and customize it according to your requirements. We provide different purification levels for REO. Usually we use silica purification with silica comb. For our preclinical grade services, however, we use silica and then oligo-DT purification in tandem. So delivering very high quality product for preclinical work. We can also perform double-stranded RNA removal, and we can customize the QC packages. Depending on the grade of the product, REO or preclinical grade, we can provide with different packages that are always customizable based on your actual need. mRNA integrity is very important for expression. Here we show an example of mRNA EGFR, EGFP mRNA, and ESP-Cas9 mRNA produced by us and some of the leading competitors on the market. And as you can see, we compare very favorably to these competitors. And that shows also in the translation efficiency. You can see on the bottom charts here, our mRNA produced by GenScript showed on the left side for the EGFP significantly higher expression levels, and for ESP-Cas9 also higher expression level compared to some of the other alternatives on the market. For poly-A tailing, of course, as I mentioned, we can guarantee and customize it according to your needs. We can produce very consistent and homogeneous poly-A tail. In this example, we produced mRNA with 120 nucleotides poly-A tail, and we compared with some of the competitors, what they produced. And you can see that we compare very favorably. On the bottom left chart, you can see the cell staining, basically for EGFP signal, the green, and then the DAPI channel for blue. And you can see that our expression is very strong. On the right side, you can see the actual measurements. You can see significantly higher expression with our mRNA compared to the alternatives. We have received so far very good customer feedback. They are very satisfied. We had feedback from Cornell University. They use our mRNA Sleeping Beauty transposase. It worked very well for them. That's the data on the very top. Another customer used our fLuc mRNA, and they received excellent expression, and they were very satisfied with the performance. That's the chart on the very right for Luc expression. And Entrada Therapeutics, they used our EGFP mRNA that in vivo in mice, and they were very happy and satisfied with the results. Some of these more popular mRNA products, we also are introducing them as off-the-shelf products. For example, the reporter GNA mRNAs, EGFP, fLuc, luciferase, and Cherry. Those are off-the-shelf products now that you can easily order. Gene editing mRNA is ESP-Cas9, Sleeping Beauty SB100 transposase, also off-the-shelf product. You can order those as standalone or in combination with other services or products. We are also starting to offer circular RNA services. Circular RNAs are a very novel molecule for therapeutics. Until recently, they were considered as aberrant splicing byproducts, and not much about their function was known. However, now there have been many papers coming out that have identified various functions for these circular RNAs. Some of the functions as miRNA sponge, protein sponge, basically regulating translation, and so on, various functions. And they're very interesting for therapeutic development because they offer very good stability, better stability than linear mRNA for longer expression duration. They offer usually higher protein expression. They re-engage the ribosomes with rolling circle translation. Also, the circular RNA LNP seem to have better thermal stability, so they're more stable and more resilient for delivery and logistics. So they're being very heavily researched right now for therapeutic use. You can see the number of publications based on PubMed search exponentially grew from a few hundred early on to several thousands per year in the recent years. So very high interest in using those for therapeutics. So a little bit more detail about circular RNA products and services we have developed. We offer two levels of services, two different quantities, micro and mini, 0.2 to 1 milligram or 1 to 10 milligram. We offer different IRS options. We offer the typical public domain CVB3. We can also customize the IRS domain for you and design it based on your specifications. We do typically reverse phase HPLC purification, so we can deliver very high purity. And we do a variety of QC measurements to ensure the integrity and quality of the product. So here is an example of circular RNA EGFP expression and analyzing by flow cytometry. You can see that the circular RNA performed significantly better than the same sequence of linear RNA, mRNA. Also, our produced mRNA performed better than what we see in the literature from other companies producing such mRNA. So you can see the data on the left. The expression level started very high, higher than the others, and stayed higher during the length of the experiment. And then the day one and total expression level were much higher. We also offer lipid nanoparticles packaging service now for mRNA delivery. As you know well, lipid nanoparticles consist of several different components, usually cholesterol, phospholipids, ionizable cationic phospholipids, and PEG lipids. And those are usually assembled into lipid nanoparticles to encapsulate the mRNA and preserve it for delivery and within the body. So here is a little bit more detail about our LNP formulation service. So we can package anything from 1 milligram to 10 milligrams of mRNA of 5 kilo bases or less. Usually this is suitable for intramuscular delivery for animal studies only at this point for our service. And we can ship it at appropriate conditions, frozen as needed, and encapsulation efficiency is higher than 80%. And we do a variety of measurements to show the consistency and reproducibility of our process. The final concentration can be customized as per your needs. Here is an example of our LNP packaged mRNA. One example comparing of GenScript formulations with a well-known competitor on the market. And you can see our formulations compared very favorably with higher expression levels. Also, here is shown on the right bottom, you can see the in vivo experiments that were also successful with injection in mice. So for preclinical studies, usually vaccine effectiveness needs to be evaluated, sometimes in vitro, looking at protein expression levels to determine how well the molecule is expressed. If it's mRNA, for example, or DNA vaccines, then usually immunity needs to be determined based on animal models, mouse models, monkey models, and so on. So cellular immunity evaluation needs to be performed looking at cytokine levels, sometimes using ELISpot or flow cytometry, humoral immunity evaluation, looking at IgG, IgA, IgM expression, neutralizing antibody expression determinations using either ELISA kits or a virus or pseudovirus, and vaccine efficacy needs to be determined. So some of the solutions that GenScript can offer for you in the preclinical stage are, we can develop the molecules for you. As I mentioned, of course, we can produce the plasmids, preclinical grade plasmid. We can produce IVT mRNA preclinical grade at very high quality and purity. We can offer some of the tools that are needed for the analysis for protein expression levels, Western blotting tools, custom antibodies for various immunoassays, custom ELISA kits for detection. For cellular immunity evaluation, we can develop peptide pools and libraries for you, and we can develop also ELISA kits for humoral immunity evaluation. And this is the summary of what we can do for mRNA vaccines. Basically, we can support you from the very early stage, from antigen identification, lead generation and optimization, preclinical studies and clinical trials. We can support your pathogen studies with sequencing products, enrichment for sequencing, PCR products and services, antigen identification and determination, basically we're looking at protein expression, immunoassays, Western blotting, ELISA kits, antigen epitope studies with custom peptide libraries. We can also support you for tumor-specific antigens with our neoantigen peptide synthesis service. For lead generation optimization, we can produce anything from the genes to be synthesized, vector construction, plasmid production, plasmid linearization, mRNA production with all the optimization that is needed, 5' capping, poly-A optimization, codon optimization, scale up as needed. And of course, we can offer similar for the preclinical studies, preclinical plasmid, preclinical mRNA, protein expression evaluation tools, immunity, humoral immunity, cellular immunity, and so on. And then our GenScript Pro Bio division can also support you during your clinical trials and manufacturing with GMP-grade plasmid and GMP-grade mRNA IVT production, as well as required GMP manufacturing analytics. So I will also very briefly go over the other solutions we offer for other types of vaccines, peptide, protein, DNA, and virus. I will touch mostly on the services and products that I haven't mentioned for the mRNA vaccines. So for example, for peptide-based vaccines, we can offer very wide range of peptide synthesis services. We can generate the peptide library services I mentioned. Typically for vaccine, the most often used ones are the overlapping peptide or alanine scanning libraries. We can synthesize neoantigen peptides for your assays with high success rate, 8 to 50 amino acids, high quality controls. We can scale up the peptide synthesis up to kilogram level if needed. We can produce it with high purity, 98%. We can keep it with low endotoxin levels, and we can synthesize peptides of up to 200 amino acids. For recombinant protein vaccines, we can do a lot of the services I mentioned already, anything from gene synthesis to plasmid purification and others. I will very briefly touch on some of the services that I haven't touched before, like our protein expression service, for example. We have very strong protein expression capabilities. We have multiple platforms. Here I'm showing only two of them. The mammalian expression platform with either HEC or 293 or CHO cells, or the E. coli expression platform. We also offer insect cell expression platform, baculovirus-based. But a little bit more detail on the TurboCHO platform, which is very popular at the moment. We have various levels of service, from very small quantities to increasing level of quantities, the high throughput, CHO Express, CHO HP, that can produce progressively higher amounts of protein. You can see on the bottom the turnaround time starting from 2 weeks or 10 business days for the high throughput to 3 weeks and 6 to 7 weeks for CHO HP for larger quantities. They can produce at very high purity and very low endotoxin levels of less than 1 EU per milligram of protein. We can also, of course, do E. coli expression very efficiently. We can start from gene sequence and finish with protein delivered in 3 to 4 weeks, performing the gene synthesis and cloning, the transformations at high throughput, the protein expression and high throughput if needed, and the protein purification, and then delivering purified protein with all the required QC documentation. And again, as I mentioned, for our product division, if you decide some of those operations you would like to perform them in-house, we can support you with for that also. We have very innovative instrumentation for protein purification and antibody purification. We have fully automated system for purification. It can purify 12 samples at a time. The samples can be up to 50 milliliters each. And each sample can contain up to 80 milligrams of protein or antibody. And typical cycle times to purify all 12 samples is 50 to 60 minutes. The instrument is programmable to accommodate different protocols and different expression tags with different purification magnetic beads. And elution volume can be as low as 0.5 milliliters. The main benefits of this instrument is increased throughput. You can purify a very high number of molecules very quickly. 60 to 70 proteins can be purified within a day. We can also save you time and labor because this system is based on magnetic beads. So magnetic beads are more resilient to contamination. So your sample don't need to be centrifuged and filtered before the purification. So you can use crude lysates, or if your proteins are secreted, you can use the cell culture inside the instrument to remove the cell debris and deliver purified protein to you. For DNA vaccines, of course, we can produce, as I mentioned already, a lot of the plasmids. We can start from gene synthesis, codon optimization. We can produce gene circle, and we can produce the plasmids that you require, and we have a lot of strong offerings in the molecular biology area. This is more detail on that part. For viral vaccines, we can basically be end-to-end provider. Again, we can start from gene sequence. We can produce all the required plasmids for your virus. We can package them in a cell line and then purify them and basically deliver the purified virus, either AAV or lentivirus, depending on your requirements. So basically, we can offer all these services and products from beginning to end for gene synthesis, plasmid, virus preparation. We also have a lot of products that are related to specifically to the virus analytics. For example, we have lentivirus ELISA kits, we have AAV2 ELISA kit, we have a lot of antibodies against AAV variants, and we also have some of the virus-related antibodies. We have benzonapirase, which is alternative to the benzonase for digesting the genomic DNA during virus preparation. And we have BSA ELISA kits and CHO HCP ELISA kits that are ready to on the shelf. For viral vector vaccines, we can be essentially one-stop solution. We can start from the very early stage with our CRO and product divisions supporting you during that stage, and then eventually switch this for your IND, clinical and preclinical and clinical studies and commercial production to our CDMO ProBio division that can produce this under GMP conditions. So from beginning to end, we can support you through the whole process. And this is my summary, my last slide. So vaccines obviously are very important for human health. According to the CDC website, more than 4 million deaths per year are prevented just due to the childhood vaccination, and many more due to subsequent vaccinations. mRNA technologies are becoming more and more popular for vaccine development, very rapid development process, as we saw the examples with COVID. And also, a lot of the technology might be transferable from one vaccine to another. Some of the issues for dealing with mRNA degradation and stability and expression, those could be translatable from one vaccine to another, accelerating the development. And GenScript can offer a complete workflow of services and products for mRNA vaccine production, everything starting from research to preclinical and clinical stage, we can support you to that either with services, with products, with research-grade services and products, or preclinical, all the way to GMP production. We can also offer very strong support for other vaccines types, such as peptide, protein, virus, and DNA. We have complete portfolios that we can support you from beginning to end. And thank you for your time, and I'm ready now to take any questions that you may have.
Thank you, Roumen, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. Okay, let's get started. So, Roumen, we have quite a few questions coming in. I'm going to start with this one. How quickly can you deliver preclinical grade plasmid?
So our preclinical grade plasmid can be delivered within 9 business days, and that's only if you order it as a standalone service. If you actually order it as part of another service, for example, our gene synthesis and plasmid development, we can also shorten the time for that, and it's not going to take 9 additional days. It will be more like a few extra days to deliver the preclinical grade plasmid to you.
Excellent. Our next question. What is the amount of plasmid that your instrument can purify per sample?
Okay, very good question. There's no real cutoff of what we can purify and cannot purify with this instrument, but the performance of the instrument, the purity and the yield is the highest in the range of 300 micrograms to 1.2 milligrams. Again, it doesn't mean you cannot purify lower or higher, it's just some of the yield or the purity may not be as high as if you're in this range.
Very good. Next question. What is the size of your mini circle vector?
The mini circle vector is very small because we have engineered as much out of it as we could. So it's only 429 base pairs.
We have time for a few more questions. Let's go with this one. Can you guarantee low endotoxin levels for the mRNA production?
Yes, we can guarantee in our mRNA production, in our IVT service, we can guarantee less than 10 EU per milligram of mRNA.
Very good. And Roumen, we're going to go ahead and wrap with this final question here. Can you guarantee the A-tailing lengths of the mRNA?
So we have multiple technologies for producing the A-tailing, and we can definitely guarantee the length, and we can definitely meet your requirements, whatever they are, 100 A's or less or more, we can definitely deliver. And we have very high consistency, homogeneity of our A-tailing. So that's what I showed on one of my slides. Thank you.
Very good. Thank you, Roumen. Now, do you have any final comments for our audience?
I just want to thank you, everybody, for attending our webinar. And again, GenScript is here to support you through your developments. If you're developing mRNA vaccine or other type of vaccine, we are here to support you with services and products, anything from research grade to GMP grade products or services. And again, if you have any more questions, feel free to contact me or contact GenScript with those questions. Thank you.
Thank you again, Roumen, for your time today and for your important research. Now, before we go, I would like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Any questions that we did not have time for today, and those submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when this is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us. Until next time, goodbye.
Thank you. Bye-bye.